Aging represents a complex, multifactorial biological process marked by the gradual accumulation of cellular and molecular damage that occurs over the course of a lifetime. While aging is an inevitable aspect of life, interventions exist to mitigate its effects by slowing the rate of damage accumulation. Rather than merely extending lifespan, the total number of years lived, anti-aging strategies emphasize extending health span, the period of life spent in good physical and mental health. This distinction is crucial as it prioritizes quality of life over mere longevity. Genomic instability is the second hallmark of aging, characterized by the progressive accumulation of DNA damage throughout life. This damage can result from environmental factors like radiation, chemicals, and oxidative stress, leading to mutations that disrupt normal cellular processes and contribute to age-related decline. DNA damage includes breaks in the double helix, errors during replication, and chemical modifications. Over time, these accumulate faster than repair mechanisms can handle, causing genomic instability. To mitigate genomic instability, individuals can minimize exposure to radiation and environmental toxins while adopting a diet rich in antioxidants. Antioxidants neutralize free radicals that cause oxidative damage to DNA. Further research is needed to develop more targeted interventions for repairing DNA damage and preventing genomic instability as we age. Telomeres are protective caps located at the ends of chromosomes. These structures prevent DNA damage during cell division by acting as buffers that shorten over time. As cells divide, telomeres naturally shorten with each replication cycle. This progressive shortening contributes to cellular aging and can eventually lead to chromosome instability. Maintaining telomere length is possible through lifestyle interventions. Regular exercise and effective stress management have been shown to support telomere maintenance and potentially slow the aging process at the cellular level. Emerging therapies, such as telomerase activators, are being developed to restore telomere length. These compounds work by activating telomerase, an enzyme that can rebuild telomeres and promote cellular longevity. While promising, telomerase activators require further research to confirm their long-term effects, safety, and efficacy in humans. Epigenetic alterations represent one of the 9 hallmarks of aging, involving changes in gene expression that occur without modifications to the underlying DNA sequence itself. These changes can affect how genes are turned on or off, influencing cellular function and contributing to the aging process through what is known as epigenetic drift. One practical approach to monitoring epigenetic alterations is through epigenetic clocks. These are sophisticated biomarkers that assess DNA methylation levels to determine biological age, which may be higher or lower than a person's chronological age. The process of DNA methylation, a key epigenetic modification, is influenced by various metabolic cycles. The folate cycle and methionine cycle provide essential methyl donors, while the TCA cycle and mutations in genes like IDH1 and IDH2 can inhibit demethylation enzymes. Epigenetic alterations also involve modifications to histone proteins within nucleosomes. These include methylation, acetylation, ubiquitination, and phosphorylation, which can alter chromatin structure and gene accessibility. To counteract epigenetic alterations, dietary interventions focusing on methyl donors like folate are recommended. Foods such as leafy greens, beans, nuts, and certain seafood provide these essential nutrients, supporting healthy methylation and potentially slowing epigenetic drift. While these approaches show promise, comprehensive longitudinal studies are needed to fully elucidate the long-term effects of epigenetic interventions on the aging process. Loss of proteostasis is one of the 9 hallmarks of aging. It refers to the decline in the cell's ability to maintain proper protein homeostasis, leading to the accumulation of damaged or misfolded proteins. This accumulation can impair cellular function and contribute to the development of age-related diseases, including neurodegenerative conditions like Alzheimer's disease. A key mechanism for countering this is autophagy, the process by which cells degrade and recycle their own components to maintain proteostasis. Enhancing autophagy through interventions such as supplementation with spermidine, a natural compound, can help preserve proteostasis. The protein degradation pathway, involving processes like ubiquitination and proteasomal breakdown, also plays a crucial role in maintaining cellular health. Studies exploring autophagy enhancement and other strategies to restore proteostasis are ongoing, offering hope for interventions that may slow aging. Deregulated nutrient sensing is one of the 9 hallmarks of aging, characterized by the malfunction of pathways that monitor nutrient levels and regulate metabolism. This disruption leads to inappropriate cell growth and reduced cellular repair mechanisms. Two critical pathways involved are mTOR, which promotes growth and synthesis when nutrients are abundant, and AMPK, which activates during energy deficits to restore balance and trigger autophagy. The AMK pathway responds to low energy states, such as those induced by exercise or fasting, by inhibiting mTOR and stimulating autophagy to clear damaged cellular components. Conversely, mTOR is inhibited by compounds like rapamycin, which blocks protein and lipid synthesis while encouraging cellular recycling processes. Caloric restriction mimetics are substances that replicate the benefits of reduced calorie intake without actually limiting food consumption. Metformin, commonly used for diabetes management, activates AMPK and inhibits gluconeogenesis, thereby improving metabolic health and potentially slowing aging. Rapamycin, an immunosuppressant drug, directly targets mTOR to promote autophagy and has shown lifespan extending effects in animal studies. The principle of caloric restriction emphasizes reducing intake of calories, sugar, starch, and protein to activate protective pathways and reduce risks associated with overconsumption. While these approaches hold significant promise for promoting longevity, ongoing research is essential to fully understand their mechanisms and ensure their safe application in humans. Mitochondrial dysfunction is one of the 9 hallmarks of aging, characterized by a decline in the energy-producing capacity of mitochondria, the organelles that serve as the cell's powerhouses. Mitochondria generate ATP through oxidative phosphorylation, providing the energy needed for cellular functions. As we age, mitochondrial function diminishes, resulting in reduced ATP production and increased oxidative stress. This dysfunction contributes to the aging process by impairing cellular metabolism and promoting the accumulation of damaged mitochondria. Restoring mitochondrial function through NAD+ boosters, such as nicotinamide riboside and nicotinamide mononucleotide, can enhance energy production by supporting NAD+ levels essential for mitochondrial metabolism. Nicotinamide riboside is a precursor to NAD+, helping to maintain mitochondrial health and combat age-related energy decline. Ongoing studies are evaluating the efficacy of these NAD+ boosters in improving mitochondrial function and potentially slowing aspects of the aging process. Cellular senescence is a key hallmark of aging where cells enter a state of permanent growth arrest but remain metabolically active. These senescent cells accumulate over time and secrete harmful inflammatory signals that contribute to tissue dysfunction and age-related diseases. To address this, senolytic drugs such as fisetin and quercetin are being developed to selectively target and eliminate these harmful senescent cells. The 8th hallmark of aging is altered intercellular communication, which involves disrupted signaling between cells that leads to increased inflammation. This inflammatory response, often called inflammaging, contributes to age-related diseases by promoting chronic low-grade inflammation throughout the body. This diagram illustrates the inflammatory cascade, showing how various cells and molecules interact to perpetuate inflammation, including cytokines and chemokines that amplify the response. To counteract altered intercellular communication, anti-inflammatory compounds such as resveratrol have shown promise in preclinical studies by reducing inflammation and supporting cellular health. However, research on anti-inflammatories and their role in aging is still ongoing, with clinical trials needed to establish their long-term efficacy and safety. The Mediterranean diet emphasizes foods rich in fruits, vegetables, whole grains, and healthy fats, demonstrating significant potential in reducing biological age. Key components include a variety of fruits and vegetables, whole grains, healthy fats from sources like olive oil and nuts, along with moderate consumption of fish, poultry, and dairy products. Research indicates this dietary pattern can reduce biological age by approximately 3 to 5 years, thanks to its anti-inflammatory and antioxidant properties that support overall health and longevity. These benefits stem from the diet's emphasis on nutrient-dense foods that combat oxidative stress and inflammation, key contributors to the aging process. Ongoing research continues to explore additional dietary patterns that may offer similar anti-aging benefits, expanding our understanding of nutrition's role in healthy aging. Intermittent fasting, particularly the 16-8 method, involves alternating between periods of eating and fasting. During fasting, the body undergoes cellular repair processes that may contribute to slowing the aging process. In the 16-8 approach, individuals consume food within an 8-hour window and abstain from eating for the remaining 16 hours each day. This pattern aligns with the body's natural circadian rhythms. Fasting triggers autophagy, where cells remove damaged components and regenerate, potentially mitigating hallmarks of aging, such as mitochondrial dysfunction and loss of proteostasis. Research indicates that intermittent fasting improves metabolic health, reduces inflammation, and may decrease biological age by approximately 3 to 5 years, according to studies like those published in The Lancet. While promising, more longitudinal studies are required to confirm the long-term anti-aging benefits and optimal implementation of intermittent fasting regimens. High-intensity interval training, or HIIT, is a powerful approach to exercise that involves alternating short bursts of intense physical activity with brief periods of recovery. This method has been shown to improve cardiovascular health, enhance mitochondrial function, and boost overall fitness, all of which contribute significantly to healthy aging. The key benefits of HIIT for promoting healthy aging include its ability to strengthen the heart and blood vessels, optimize cellular energy production through improved mitochondrial performance, and enhance overall physical fitness levels. To illustrate HIIT in practice, here is a sample workout guide showing common exercises performed in short, intense intervals. HIIT's cardiovascular benefits are particularly noteworthy, as regular sessions can help delay age-related declines in heart function and reduce the risk of cardiovascular diseases. Research data demonstrates how HIIT can positively impact key health metrics over time, supporting its role in maintaining vitality as we age. Ongoing research is exploring the precise amounts of HIIT needed for maximum benefits, with recent studies indicating that this form of exercise can reduce biological age by 3 to 5 years, making it a valuable component of anti-aging strategies. Senolytics represent a promising emerging therapy in the field of anti-aging research. These compounds selectively target and eliminate senescent cells, which are dysfunctional cells that accumulate with age and contribute to inflammation and frailty. One prominent senolytic is fisetin, a natural flavonoid found in fruits and vegetables. Fisetin works by inducing apoptosis in senescent cells while sparing healthy cells, thereby reducing the inflammatory burden associated with aging. Senescent cells produce senescence-associated secretory phenotype, or SASP, which includes pro-inflammatory cytokines that can harm surrounding tissues and promote further cellular aging. Another effective senolytic is quercetin, often used in combination with fisetin. Together, they help break the vicious cycle of senescence, where stress leads to more senescent cells, amplifying inflammation and tissue damage. Clinical trials have shown encouraging results. For instance, a 2024 JAMA study indicated that senolytic treatments reduced frailty by 20 to 30% in older adults, improving mobility and overall physical function. This visual comparison highlights the difference between normal cells and senescent cells, emphasizing how senolytics can restore cellular health by clearing these problematic cells. Nicotinamide adenine dinucleotide, or NAD+, is a critical molecule for cellular energy production and DNA repair. As we age, NAD+ levels decline, which contributes to various aging hallmarks. Emerging therapies focus on NAD+ precursors that can replenish these levels. Two key precursors are nicotinamide riboside, NR, and nicotinamide mononucleotide, NMN. These compounds can be converted into NAD+ within cells, helping to restore metabolic balance. Clinical trials, including recent studies in 2025, have shown that these boosters improve mitochondrial function and reduce age-related muscle loss in participants over 60, potentially slowing sarcopenia. While NAD+ boosters are available as dietary supplements, more research is required to confirm long-term benefits and safety. This emerging therapy is still in the testing phase. CRISPR-based gene editing tools are being used to edit longevity genes in animal models, extending lifespan by up to 20% in some studies. Human applications are in early stages, with trials focusing on epigenetic clocks to measure biological age versus chronological age. This is still in the early stages, with regulatory hurdles and ethical considerations remaining key challenges. Artificial intelligence is transforming anti-aging research by rapidly analyzing vast biological data sets. AI algorithms identify patterns in molecular interactions that would take humans years to discover. This technology is accelerating drug discovery through predictive modeling and machine learning techniques. AI-designed molecules targeting autophagy induction are currently undergoing preclinical testing. These compounds aim to enhance cellular repair mechanisms, potentially slowing age-related decline. Precision longevity involves tailoring anti-aging interventions to an individual's unique biological profile. This approach uses specific biomarkers to guide personalized strategies for slowing the aging process. Interventions are tailored based on individual biomarkers, which are measurable indicators of biological processes. These markers help identify areas where aging interventions can be most effective for each person. Blood tests measure inflammatory markers, such as C-reactive protein and interleukin 6. Elevated levels of these markers can indicate chronic inflammation, which accelerates aging. Monitoring these helps customize anti-inflammatory interventions. Wearable devices, such as the Oura Ring, continuously track physiological data. These devices monitor sleep quality and heart rate variability, providing real-time insights into an individual's health patterns. This data contributes to personalized aging metrics, creating a comprehensive profile of an individual's biological age. By analyzing these metrics, healthcare providers can recommend targeted lifestyle changes or therapies. While precision longevity shows promise, further research is required to fully understand the long-term effects of these personalized approaches. Ongoing studies will help refine these methods for broader application. Brian Johnson, a 47-year-old tech entrepreneur, has developed the Blueprint Protocol, a comprehensive approach to slowing the aging process through over 100 daily interventions. The Blueprint Protocol incorporates over 100 daily interventions, including peptides to support cellular health, regular sauna sessions for detoxification, and a strictly plant-based diet rich in nutrients. His exercise routine, detailed in his anti-aging workout guide, focuses on high-intensity movements to maintain fitness levels comparable to those of much younger individuals. Johnson claims to have reduced his biological age to 42 years, as indicated by true age epigenetic tests, along with enhanced energy levels and improved biomarkers. While his 2024 book Don't Die documents these personal results, scientific validation requires long-term studies. Moreover, individual responses to such protocols can vary significantly, and these results may not be representative of the general population. Okinawa, Japan, has the world's highest concentration of centenarians, with a rate of 50 per 100,000 people compared to the global average of just 8 per 100,000. Located in southern Japan, Okinawa's unique geography and culture contribute to this exceptional longevity. The traditional Okinawa diet is low in calories and rich in fish, vegetables, and soy products, which provides essential nutrients while maintaining healthy weight. Genetic factors also play a significant role, with studies identifying longevity genes like FOXO3A that are more prevalent in Okinawan populations. Research continues to study these centenarian populations to better understand the complex interplay between diet, genetics, and lifestyle factors that contribute to exceptional longevity. David Sinclair, a prominent Harvard geneticist, argues that aging should be viewed as a treatable disease rather than an inevitable process. Sinclair strongly advocates for NAD+ boosters, which help restore declining cellular energy levels that occur naturally with age. He also promotes cold exposure as a beneficial stressor that triggers hormesis, strengthening the body's defenses against aging. Supporting his views, Sinclair references studies showing that mice treated with these methods lived up to 50% longer than untreated counterparts. Although these results are encouraging, it is crucial to note that extensive human clinical trials are still required, as the current evidence primarily comes from animal models like mice. Looking ahead, Experts in the field of longevity research predict a phenomenon known as longevity escape velocity. By 2030, life expectancy may increase by more than 1 year annually, potentially reaching 150 years or more through continued advancements in geoscience and biotechnology. This concept, popularized by futurists like Ray Kurzweil, suggests that scientific progress will outpace the aging process itself. Current trends include declining rates of age-related diseases like dementia, driven by preventive strategies and early interventions. However, significant challenges remain. Access to advanced therapies may be limited to wealthier populations, exacerbating social inequalities. Ethical considerations include potential overpopulation and the need for equitable distribution of life-extending technologies. In conclusion, slowing the aging process requires a multifaceted approach that combines science-backed lifestyle habits with emerging therapies. While individual results may vary based on genetics and adherence, consulting with healthcare professionals is essential for safe and effective interventions. Self-experimentation can be risky, and it is important to proceed with caution and expert guidance.